the Almighty and His Holiness One. At the onset, I would like to invite my classmate Arpita Uday Nayak to sing the invocation. To introduce our guest and welcome the gathering.
Very good morning to one and all. Dignitaries, guys, and students. At the outset, I offer my sincere pranam to His Holiness Jagat Guru Sri Shivaratri Deshikendra Swamiji, and I feel honored and privileged to be here today proposing the welcome address and also to introduce the guest today. The guest here today is very special to all of you, but more special to me because we have been classmates since, 20, 25 years we know each other. So it's always a pleasure to associate back with him whenever he is here. He is Dr. Sandeep Prabhu, a professor of immunology and toxicology and Head Department of Veterinary and Biomedical Sciences, the Pennsylvania State University, USA. Just to brief about his activities, his research activities and his future at the university. The current focus of his laboratory at Penn State University is on the redox regulation of pathways of inflammation and resolution for nutrients, specifically the selenium and omega-3 fatty acids, that act through the production of indigenous lipid mediators. His current projects are focused on bioactive metabolites derived from these nutrients and their effect on immune homeostasis in models of leukemia and other inflammatory diseases, both bacterial and parasitic infections, which involves gut dysbiosis. So, utilizing the power of mass spectrometry based metabolomics and proteomics in conjugation with molecular, immunological and biochemical methodologies, they have discovered novel bioactive lipid metabolites that can selectively activate pathways of resolution of inflammation by selectively skewing the phenotype of macrophages. These mediators also target leukemia stem cells. All these projects were successfully carried out by many of his graduate students and postdoc fellows. Thus, one of the goals of his laboratory is to also train the next generation scientists. Many of you might become one if you are competent enough. Many of his former postdocs and students have been successful in academia and industry and have established themselves well. In his role as department head, he has focused his energy towards recruiting highly talented faculty, particularly those from the underrepresented groups. He has also mentored many junior and mid-level faculty towards tenure and promotion. I extend a warm welcome to him and request our beloved principal, Dr. Kibade sir, to present him a bouquet and thank you very much, sir. We have with us today Professor Kibade, the Principal, Dean Engineering and Technology of our university. It's a pleasure to always have his support and enjoy his support being here and encouraging us with all his support always. Thank you very much. It's a privilege to always welcome him on all occasions like this. A support like his is very essential and he is always ready to give it to us. It's also a special occasion for all of us today in the department. I was just talking to Dr. Nagendra Prasad. One of the occasions wherein another faculty of our department has occupied a higher position in the university, and this is the very first occasion we have to not only welcome him but feel grateful also. So we have with us today a very good examination, and we are proud of you. Thank you for pulling out time and being here. Welcome. Yes, another great personality we have today with us, Dr. Manjunath Swami, is head of the department, and as you know, him always a very prominent researcher in the department, contributes towards motivating students, towards research and other activities in the department. Thank you for joining us today and supporting us. So it was a pleasure having the honor of welcoming the dignitaries today. Thank you.
Thank you, Prasad sir, for introducing the guest and welcoming the guests that I have. I'd also like to welcome you for the debate. I'd like to invite Dr. Sandeep Prabhu on the dais to address the gathering.
Thank you very much for the kind introduction, invitation, and putting this thing together. Indeed, a privilege and an honor to be here to talk to you all, share what we do in my lab and at our university. But before I do the scientific talk, I wanted to recognize the importance of what your professors are doing for you. This is really very nice for you all at this particular stage in your life to get some exposure, because when I was a student, we never had these opportunities. And so you should be thankful to your principal, sir, and Dr. Prasad, and Nagendra Prasad, and Swami, and others. Really wonderful to have, faculty from other institutions, particularly abroad, come and share their, research. Because even though whatever I'm going to tell you may be out of your realm of what you do, but you never know where it might be used. And particularly for biotechnology students, when you go into an industry, when somebody is talking to you, some meeting or something about inflammation, you will, you would have some ideas, and those ideas are enough sometimes to take you to the next level. So such things are very important. I also tell my students to go attend talks from computer scientists or from biomedical engineers, because they totally think differently. We have a single track mind, we think differently. Whereas computer scientists, engineers, my wife is an engineer, is a product of this great institution herself, thinks totally differently. And I feel I am a, sort of a fool when I talk to her because she thinks logically and I don't think logically. I think in a different manner. But it helps when you listen to talks, something that is outside your topic, it actually helps. So, I would like to thank you all for being here and listening to me. And if you have any questions, I'd be happy to answer about anything regarding careers or prospects and others. Because as Professor Prasad said, I was here in the same room about 5 years ago, giving a young program. Thanks to Dr. Hemant. We did a very nice program together on personalized cancer medicine. It was wonderful. I enjoyed being here. I had never taught in India. So this was a very nice opportunity for me to teach students. We went through a lot of bioinformatics, we did a lot of stuff. We designed some CRISPR-based targeting and all that, which was very nice for students, I think. So I really enjoyed and interacting with you all. So good luck. You're in a great institution with great faculty. Make best use of it. Be focused. You'll all do well. Thank you.
Thank you, sir, for addressing the gathering. And I really nice to right here just to be multidisciplinary. We hope we all will do that in the future. Thank you. Now I'd like to call upon our dear principal, Professor S. Shivari, to give the presidential remarks.
Very good morning to all. Seeking the blessings of the community, respected speaker, Dr. Sandeep Prabhu, in charge of the department of Convention Sorry, Technical Agindra Prasad, Dr. Prasad is organizing this program, Dr. Hemant, Lakshmi Madam, and my dear students. I'm not a biotechnologist, but I'm thinking from morning. Yesterday he told me, sir, you have to address the gathering and what to address. I have discussed from last 30 months. I am discussing with the sir to tell something about related to the biotechnology. I'm not a biotechnologist. Just whatever something, just within 2-3 minutes, I will address you little bit what I know regarding the biotechnology during the COVID period and what is the importance of the biotechnology, especially the applications of the biotechnology in the field of the biochemistry, in the field of nanotechnology, in the field of information technology, especially in the field of agriculture. What is the importance of this biotechnology? Now the scenario, in the global scenario in the 21st century, is the global changer. The biotechnology field is the global changer for the 21st century regarding the economy. Now, almost especially from the last 2 years, the pharma sector, health sector, and especially biotechnology, this field got a lot of importance during the COVID period. And before that, we are not much familiar about these sectors. Importance of the sector as well as the economy and the growth totally directly it will help to the economy of the country, and how it will be useful to the daily life, not only for wearing what clothes we are wearing, how to wash it, and even for the food processing system. Many of the things, how these applications are using. Most of the things we are not aware of it, but we are doing daily. We are using it, but we are not aware of it. But how it can be used in the properly. But with these few words only, because I don't know much about it to talk about the biotechnology. Remaining things that Sandeep Prabhu will address you, but only things that student have to get the benefit of this program. And sir is having 25 years plus of experience. They have a lot of knowledge. They are practical, theoretical, and this program was organized for you people by the Department of Biotechnology. First, I will take this opportunity to congratulate all the faculty members and in the absence of your head of the department, Dr. Sanjay, and all of you have to attend this program. Do you have any questions regarding all this? Informed you regarding about the career prospects and the subject, out of the subject also, when it is going to help you. It is good to know when it is going to help you. But right now, we'll think out of the curriculum, but always we will focus on the point of examination point of view. But always you have to think out of the box and you have to study the subjects which is useful in our future. With these few words, thank you once again for giving me an opportunity. Wish you all the best.
Thank you for the presidential remarks, sir. Thank you for encouraging us to do something more in the near future. Now I'd like to call upon Dr. V.P. Hema to deliver the vote of thanks.
Good morning, all. Most of the things in our department happen with short notice. First of all, I would like to thank Professor Sandeep Prabhu for accepting our invitation with a short notice. Thank you for being here. I'd like to extend warm gratefulness and thanks to our principal, sir, who always supports us in this. Thank you, sir. And recently we have one of our colleagues taking up the higher position of being Deputy Controller of Examination. I'd like to thank Professor Nagana Prasad for joining us. I would like to extend my thanks to our beloved HOD who could not join us today, but he has always been supportive and encouraging to all of us. Thank you, sir, in your absence here. I'd like to thank our in-charge HOD, Professor SNS, who is always join us and whatever we do. Thank you, sir. Not just all these, we have other people who have helped us, all my colleagues, Ms. Lakshmi, Pallavi ma'am, Kritika, everyone who have been always supportive when we do any program in our department. It is a teamwork, so I would like to extend thanks to all of them, even to the non-teaching staff, Shashi, Bhaskar, Rigana, who are not here, but they're always our backbone. I also like to thank our student volunteers who are always willing to help us in any of the functions we do. I'd like to start with Arpita for readily agreeing for invocation. Rajat for agreeing to do the MC part. Thank you, Rajat. And Aruna for taking all the pain and helping me in getting the pins done. Thank you. Not just those three. Those all who have joined us here, you all are our guests. Because of you, we have this function. Thank you one and all for joining us. Thank you.
ये पकड़ ये ले पकड़ ऐसे
begin by telling you about one of the projects in my lab, and that is on diet and what is called resolution of inflammation. I'll tell you a little bit about that later. And I'll make a case for an element, okay, which you may have read about in your PUC or maybe in your middle school when you studied the periodic table. Right? How many of you remember the periodic table? Right? Only few. Okay, good. Yeah, so I'll tell you a little bit about one of the elements called selenium. And you will appreciate how important an element is. You all know iron is very important for your metabolism. Calcium is important. Magnesium is important. But they all act differently. And I'll tell you what selenium does. So my lab works on, as I told you, what is called resolution of inflammation. And what this means is, whenever there is inflammation, if you get exposed to pathogens, like for example with COVID, right? One of the things, pathologies that resulted from COVID was essentially because of inflammation. It was massive, exacerbated inflammation, and that inflammation did not resolve in a timely fashion, and that's what caused multi-organ failure. Okay, so if you look at the COVID patients, took their serum or whatever and then did analysis for cytokines, the levels of cytokines were very high, and that signified inflammation. Similar to that, if you get exposed to toxicants, environmental toxicants, like for example lead, lead or mercury or something like that, or pesticides, you actually can also, it can lead to inflammation, and that ultimately can result in disease. Similarly, nutrition deficiencies will also cause this. So, my lab is looking at many different nutrients, but of late, there are a couple of them that we are interested in, and one is selenium. The other one is omega-3 fatty acids, which are present in fish, or others like flaxseed and other things, which is now very important. All of you recognize flaxseed oil or flax seeds are very important. So we also work on it. And I don't work on just the nutritional part, in the sense we don't do what is called feed and feed type work. We look at more molecular mechanisms. Okay, I try to understand at the level of the cell what happens. If you eat whatever you eat, is it good for you? And if it is, how is it working? I'm not interested in mechanisms. So we work on multiple different diseases as shown there. One is endotoxemia. It's nothing but sepsis. You may have heard of sepsis. And then we work on infections, helminth infection. I'll show you a little bit on helminth infection and IBD colitis. Have you heard of this? IBD and colitis? It's a massive inflammation of the GI tract. Today I'm not going to talk about cancer, even though leukemia is something that is very close to my heart. And there is something called anemia of inflammation. I'm not going to talk much about that. So I'm going to talk about IBD colitis and infections, mostly related to the brain. So let me tell you a little bit about selenium. So this is the periodic table, the threaded periodic table, right? So selenium is in the family of oxygen sulfur. You can see here, right? Group 6A. And this is the element. Now, it has properties of a metal as well as a non-metal. So that's why we call it a metalloid. Okay. And this was discovered about 200 years ago by a Swedish biochemist, physician, by name Jan Jakob Berzelius. Okay. And Berzelius was a physician, but was interested in basic research. So he not only discovered selenium, but he also discovered tellurium. He discovered a few others as well. And so about 5 years ago, we all met in Stockholm, in Sweden, where Berzelius was, and we had a nice meeting or a conference to commemorate 200 years of Selenium Discovery. And this is a picture of Berzelius in a park in downtown Stockholm, where you see the statue of Berzelius and I was fascinated by this because not in many countries you will see scientists on a pedestal. It's mostly kings or politicians or public figures. Sweden is a little different. They have a lot of scientists in their parks. Maybe the scientists went to parks. Anyway, so that is Berzelius's picture, statue there. Sorry. And then this is his lab. This is exactly 200 years ago he discovered selenium. We were able to open this lab and go in as well. It's a small room, nothing sophisticated. 200 years ago he didn't have a mass spectrometer, he didn't have a flame photometer, he didn't have any gadgets. He had very simple equipment, and based on the reactivity in acids and bases as well as their nature when you expose it to a flame, what kind of color they emit. Actually, Berzelius identified many elements. You know, in addition to Mendeleev, you remember Mendeleev's work? Russian biochemist. So Mendeleev, in addition to Mendeleev, Berzelius actually was also a key figure in putting the periodic table together. Not many of your textbooks say that, but actually Berzelius was a key figure. So this selenium is a trace element, and we often refer to it as part of being a 21st amino acid. Now you know there are 20 amino acids, right? This is part of the 21st amino acid. Now I'll tell you, it may be confusing because I'm telling you it is an element, then I'm telling you it's an amino acid. So it may be confusing, so I'll resolve that in a bit in the next few slides. But what happens is, when there is deficiency in selenium, you see a disease called cardiomyopathy. What cardiomyopathy is nothing but, there is massive inflammation in the heart and people die. And this, you know, nobody really correlated that to selenium deficiency. They knew people were dying in parts of China, parts of Scandinavian countries, including Sweden, Nordic countries, parts of Tibet, parts of UK, parts of New Zealand, all are selenium deficient. And people had these cardiomyopathy. And only when somebody was looking at the soil, this was I think was a geologist who was looking at the soil and, you know, making all the estimations of what is there in the soil, he saw there was no selenium. So he reported that there was no selenium, and it took some nutritionists to connect the two dots. So talk about interdisciplinary science. You can see how interdisciplinary science is important. So cardiomyopathy is also called Kishan disease, and there is also another disease called osteoarthropathy or Kishan disease, and both of them happen because of selenium deficiency. And then later on, it took some molecular biologists to really understand what happens during selenium deficiency. Turns out, all of us have a cardiomyopathy, a virus called Coxsackie virus. All of us have a non-cardiotoxic strain. Most of us are exposed. It stays that way, nothing happens. However, when there is selenium deficiency, that Coxsackie virus in you, which is non-cardiotoxic, mutates, and it becomes cardiotoxic. And when it becomes cardiotoxic, that's when it causes cardiomyopathy. So if you have selenium deficiency, that mutation happens, you make it really virulent. But then, if you add selenium, it mutates back, and that's how it treats. You can treat cardiomyopathy. So this we did not know until a molecular biologist actually looked at this and showed the results. In addition to these two diseases, there are also other diseases such as some of the cancers, preeclampsia in women. You may have heard this. there is high blood pressure, and it causes a lot of problems during pregnancy. And then hypothyroidism is very common nowadays, hypothyroidism, because not just because of iodine deficiency, it is because of selenium deficiency also. I'll tell you what the connection is. And this is not just restricted to humans, but also animals have this. So, let's go to some molecular biology. Okay. You all know how protein synthesis occurs, right? And this is your codon chart. You've all seen this chart, right? All the different codons and so on. Now, if you see, there are actually 3 stop codons, correct? And one of them is UGA codon. You know that is a termination codon, right? I'll tell you, UGA is not always a termination codon. That many of your textbooks won't tell you. Actually, UGA can also code for something. Okay? This is where we started rewriting molecular biology. The central dogma of molecular biology, we started rewriting it. And what we found was that UGA codon could code for a special amino acid. And that special amino acid turned out to be selenocysteine. Okay? You know cysteine, right? So instead of the sulfur, you have replaced it with selenium. Now, if you see how this happens, here is the complete, schematic. So, if you take selenium in your diet, it gets converted to something very similar to hydrogen sulfide, but instead we call it hydrogen selenide. Instead of H2S, we call it H2SE or HSE minus. And then it is phosphorylated by an enzyme called selenium phosphate synthase to this selenium phosphate. Now that selenium phosphate is used to charge a tRNA that has serine in it. So serine, you remember, is a hydroxyl containing amino acid, and that hydroxyl group will be now replaced by selenium. So this tRNA now becomes selenocysteine tRNA. That tRNA, the selenium charging of the tRNA actually happens on the tRNA itself. Unlike other amino acids, they are synthesized and charged. Here the charging occurs on the tRNA. So that's why it's different. Now, once you have the selenocysteine tRNA, it will recognize the UGA codon in some transcripts, not all mRNA, very few transcripts. And that only happens when you have multiple UGA codons. Okay, here it is one UGA and then there is another stop UGA codon. But then there is also a hairpin loop structure, a secondary structure in the 3' untranslated region of the mRNA. You know mRNA has a 5' UTR and a 3' UTR. In the 3' UTR, you have this element here called the selenocysteine insertion sequence. That makes some proteins bind to it in the ribosome and then recognizes the UGA to incorporate selenium. And then when the ribosome moves to the next stop codon, that is the real stop codon, it stops. Okay? So this is where this particular element is different. Because if you consider iron, magnesium, copper, calcium, any of these elements, metals, trace elements, they don't work this way. They all bind to proteins non-covalently. Here, it's a covalent function. Okay? Now, you can ask me, what happens if people don't take enough selenium in their diet? What happens is, the ribosome, the translation is occurring. When it hits this first UGA codon, it stops because there is no tRNA. So it stops. The peptide that is being synthesized comes apart. It undergoes degradation by ubiquitination. And then the RNA also undergoes degradation by a process called nonsense mediated decay. Okay? This is a process where RNA gets turned over. It's a very important process for RNA to be turned over. Okay? That we have all evolved with. And that particular mechanism is functional here. So if there is no selenium, the RNA gets completely degraded, and then the protein also gets degraded. So you won't get any proteins. Now you can ask me, what kind of proteins are made? So if you do a bioinformatic study and look for homology of transcripts that complete not just the ORF, open reading frame, but actually the entire transcript with the 3' and 5' UTR, you can look for this C-KISS element. And if you do that to the entire human genome or mouse genomes or whatever genome you want to, even bacterial genomes, you will see it brings up about 25 different selenium proteins in humans. So we all make all these selenium proteins. Okay, I don't want you to memorize all this, but remember there are several selenium proteins, and some selenium proteins are important in the sense we know what they do, and some selenium proteins we have no idea what they do. Particularly this lower end of this chart, we don't know what these selenium proteins do in the system. We have some ideas, but again, it's not completely clear. Whereas there are other selenium proteins here called GPX1 and GPX is such called glutathione peroxidase. There are proteins that are important for redox signaling. Then there are proteins important for thyroid hormone metabolism. See, this is where I was telling you about hypothyroidism. So you know thyroid hormones. So there is a precursor thyroid hormone called T4, tetraiodothyronine. That undergoes a deiodination and you lose one iodine atom and it becomes triiodothyronine or 3 iodines. That is the biologically active form. So if you don't have selenium, you will have very high T4 but low T3. So if you take T4 to T3 ratio and combine it with TSH levels, you actually see in hypothyroidism patients, you have very low T3, high T4. Okay. So what they do for hypothyroidism patients is they give them iodine and they also increase the selenium levels by supplementation so that you can get more T3 levels. Okay. So, and then there are some important selenium proteins. I would like to highlight a couple of them, particularly one where this is very, very interesting from a molecular biology standpoint as well, because all these selenium proteins here have one selenocysteine in their active site. They're all enzymes, so they have an active site and there is one selenocysteine. Whereas this particular selenium protein called SELP or SEPP1 has 10 different selenocysteines. So you can imagine with only one tRNA and UGA at different places. So this means if you look at the transcript of this mRNA of this, it has multiple UGAs, so multiple stop codons. So what you would say is, if you just looked at it, you would say, oh, this is a protein, big protein, but then there is UGA right here in the N-terminus, so it will form a small peptide. Actually, it incorporates selenium, and then it goes forward and incorporates 9 different selenocysteines. Okay? So if you look at the transcript of this, there are 2 selenocysteine, this particular hairpin loop structure, there are 2 of them. And the 2 of them each one is responsible for 1 of these and 9 of these. So one selenocysteine sequence is doing for 9 in one case, whereas in the other one it is doing only 1. So we really don't understand how this is happening. So for a molecular biologist, this is a very interesting problem. Okay, how it distinguishes on the ribosome itself. So that's something a lot of others are working on it. But my lab focuses on what happens when you lack these selenium proteins. So one of the diseases I was very interested in because of many reasons, and one of the reasons is my postdoc suffered from this disease. And it is very hard because I saw him suffering. He had to be given, he lost about almost 40 kgs. 40 kgs, imagine losing 40 kgs in a span of about 2 months. And he became almost like bare bones. And I was scared to see him. He was in the hospital, and he didn't have anyone with him, so I was his caretaker. And the doctors diagnosed it as IBD colitis, and then they were trying to somehow see if he can be treated. So one of the things they do is they treat it with an anti-TNF antibody called Remicade. It's a TNF alpha neutralizing antibody. Only problem is you can't keep giving it. You can give it once a month, and you have to keep this person in an isolated room, because one of the problems is, as you know, TNF alpha is a pro-inflammatory cytokine, but it is also important to keep you safe from mycobacterial infections. If you don't have TNF alpha, you could have tuberculosis. Okay, so he had to be kept in a room and everything, and and then there is massive blood in the feces or in the bowel movements, and then you can't eat anything, and it's really bad. So when they gave him TNF alpha, within 1 day we saw a lot of improvement. But within about 2 days, he started going back again. So then what they did was they said, we can't help it now, so we will go through a resection. So what they did was they did what is called colectomy. So it is a 3-step surgery. It's really bad. First is they take out part of the colon or entire colon, depending on what the surgeon will see when they open the patient. Depending on the inflammation, it could be ascending or descending colon. Then they took off the colon. Then what they did was they put a colostomy bag. And after that, they had to re-anastomize, which means they connected the small bowel or small intestine to the anus straight. Okay, it was a 3-step surgery. It took him almost 6 months to go through this, and it was very bad. So when this was happening, I was really thinking, because inflammation is a major thing there, issue, and IBD is totally incurable. There is no cure for IBD, and we see throughout the world we have over 5 million people with IBD, and in the US itself we have 1.2 million. Okay, and there is no treatment, and even if there is treatment, it costs about $15,000. Now imagine times 76. You will see how much it is. It is impossible to, even with insurance, it's tough. So an annual direct cost for all patients with IBD is in the billions of dollars. So it's not treatable. You need something else. And there are treatments such as use of anti-inflammatory steroids, immunosuppressants, but they're not very effective in all patients. Only about 40 to 70% respond. 40 may go into remission, but the rest, 60%, will have the disease again. And it is really hard, because what starts as colitis can become cancer, colon cancer. Okay, so we decided to look at it, and there is a lot of literature where patients with small bowel resection, that is small intestine resection, appear to be at the risk of developing selenium deficiency. So this is another problem. So if you do resections of part of the colon, part of where the colitis is, it could be in the small bowel or large bowel, you will now have secondary problems. You can't absorb things properly, so you will have to be given parenteral nutrition, like through IV or something like that. How many can afford all that? Can you imagine, instead of eating every day, you have to stick yourself with a line, IV line type thing with nutrients? And that's how you are going to be fed for the rest of your life. It's very tough, and that leads to more deficiency problems. And so, and people with colitis or Crohn's disease, are part of a similar thing to colitis, and on enteral nutrition, have deficiencies of selenium. And what we have seen is the SEPP1, the protein that I told you, that is inversely correlated with the development of IBD and colorectal cancer. So people with low levels of SETP1 have higher incidences of cancer. Okay. So we have some data showing that SETP1 protein is critical for protection. So we decided to test all this in a mouse model. And this is my collaborator and my mentor also, Dolph Hatfield at National Institutes of Health in Washington, D.C. And Dolph worked with, I don't know, you all know probably Marshall Nirenberg. You know Marshall Nirenberg, who got the Nobel Prize with Corona, okay, for genetic code. So Dolph worked with Marshall Nirenberg as his key scientist on the Nobel project. Okay, that got him a Nobel Prize. So Dolph was kind enough. He sent us these mice. These are genetically modified mice. And these mice lack the gene for that tRNA that I was studying that makes selenoproteins. So these mice have been engineered in such a way that when you cross them to any recombinase proteins, you can delete that particular gene in any cell of your choice. Okay? If you are interested in a cell type like macrophage or monocyte, you can use this particular mouse strain called lysine 3, and it will delete selenoproteins in all those cells that express lysine 3. If it is epithelial cells, we use what is called Willen 3. So similarly, with heart, you can use a certain creed. With neuronal cells, you can use a certain creed. So using this model, we can actually now ask questions as to which cell type and what kind of selenoproteins are important. So using this model and a wild type mouse as a control, we decided to ask the question, if you put mice on different diets containing increasing amounts of selenium, from totally zero selenium to adequate levels of selenium, 0.1 ppm of selenite, supplemented at 0.4 and very high levels, and about, keep them in the diet for 8 to 12 weeks. And then you subject them to some kind of inflammation in the gut by giving them a chemical called dextrin sodium sulfate in the drinking water. So they develop colitis, and I will show you some pictures. Or infect them with a parasite like worm. I'm sure you've all heard of worm infections. Okay. Now those worms, you could also do it here. Or infect them with a bacteria. What happens? Because you may eat some contaminated food and you will have a lot of stomach upset. That is essentially the bacteria is going into your gut, dividing and causing a lot of dysbiosis and inflammation. So we decided to look at that. So in this DSS colitis model, it can produce a lot of acute and chronic disease. And what it does is, it takes off, see this, this is an acute colitis model, and you can see there is, the epithelial layer in your intestine is completely gone. Okay, your epithelial layer has mucus. Okay, and if you remove the mucus is there for protection, but if you remove the mucus, your epithelial layer now is susceptible to any kind of inflammation. So it's like essentially taking this wire and shaving off the insulation. Now we have live wire. Okay, it's dangerous. So something similar. So when we subjected them to selenium deficiency and treated them with DSS for 5 days and put them in water, you can see that ones with high selenium actually resisted change in the body weight compared to the low selenium levels and the deficient ones. They also survived less, whereas the ones with high selenium or selenium supplementation, nothing happened, they all survived. And if you now look at the colon, one of the indications is, so this part is the colon, okay? This is the large intestine and this is the cecum, small intestine. So if you see the colon, you can see changes in the length, correct? So what happens is when there is inflammation, colon shrinks. And you can see the difference here. Can you see the difference? Right? So colon length, was also short in deficient ones, and then there was a lot of stress. And when you take sections of the colon and see, you can see this is how a typical colon should look like. Very nice structures, like the villi in the intestine. That helps in absorption and all that, increases the surface area, just like in your lungs. So if you see that in this selenium deficient ones which were highly inflamed, you can see there is no structure at all, they have lost it. And you see this basal membrane, it is thick, which means there is a lot of inflammation compared to this. They're all taken at the same magnification for comparison. Okay, so you can see progressively how it changes as you just increase selenium levels. Okay, in the mice, you can see how it changes very nicely. And then we looked at inflammation, and you can see, you can look at TNF alpha, interferon gamma, IL-1 beta, all those can actually decrease as you go up this way. Okay, at the level of RNA and protein potential. So as we were doing this, we found some papers where they had also shown, in addition to those cytokines, there was increase in other lipids, and particularly lipids that cause inflammation. For example, if you get infected with a virus or bacteria, you will have fever, correct? Do you know why the fever is because of? Anyone? Why do you get fever when you have an infection? Yes, prostaglandins. Now, which prostaglandin are we talking? You're right. So it's increasing prostaglandins, and one prostaglandin that helps in fever generation or febrile response, what we call, is this prostaglandin called PGE2. It's a prostaglandin E2 that increases in your serum very quickly and increases temperature. Okay, because there are many neurons that have this receptor for this prostaglandin. They get activated, it tells the brain you should, increase the body temperature. So you get increased body temperature. It's actually a good response to have fever. Okay, just like, sorry, I kind of digress in between, but this is very important because you need that fever to actually help in the production of antibodies. Similarly with COVID, when you were given vaccines, you may have read not to take anti-inflammatory agents, non-steroidal anti-inflammatory, particularly ibuprofen or naproxen, or even aspirin to that extent. Because if you take those, you will not get enough of PGE2, I'll tell you in a bit, and then you will not make antibodies. So no matter how much vaccine you have taken, you will never make any antibodies. So you may be under this false impression that I have been vaccinated, so I'm safe, but you are probably not. Okay? You are not making any antibodies. So it's a double-edged sword. We call it a double-edged sword. And that is because PGE2 is important. At the same time, too much of PGE2 is also bad because it causes fever and inflammation. So we saw that in patients with IBD, there was selenium deficiency, there was increased PGE2. And more importantly, there are some patients who go into remission, which means they actually can decrease their colitis and they actually can be treated. In those patients, it's a small fraction, that 40% I told you. Those patients who are in remission of disease, there was another prostaglandin that went up, called PGE2. Now, we had done a lot of work in this area, but not from a colitis standpoint, but from a totally just a sepsis standpoint. And we were monitoring the enzymes that were responsible for making these prostaglandins. And those enzymes are shown here. Okay? One is called prostaglandin E synthase, this one, which makes PGE2. The other one is called thromboxane synthase, or makes thromboxane. The other one called prostaglandin D synthase, which makes PGD2 and 15-deoxy-PGJ2. A lot of PGs, okay? And these are all from the COX enzyme. This is the target of your non-steroidal anti-inflammatory drugs. So imagine if you take NSAID, it will cut off the pathway here. Nothing will come out of this. So what it tells you is taking NSAIDs may be not so good in long term. Short term it may be good, but if you take it on long term it may not be good. And we have some literature on that. So what we did was we took just simple macrophages, treated them with different amount of selenium, as you can see here, and stimulated them with LPS, which is nothing but a bacterial product, lipopolysaccharide, and monitored all the expression of different proteins, all these proteins, by Western blot. What you see here is, as you start increasing selenium, some of these enzymes switch off. See this, thromboxane switches off at 100, somewhere between 50 and 100. PGE synthase stops same, but then PGD synthase starts to express. So it's like pressing different buttons. You turn off few, turn on few. Okay, but this is happening in a live cell. So we became interested and we decided to look at what is going on. We see that this has been published in many different papers, and after many papers we have this beautiful picture. Okay, very simplified picture. And what it tells you is when you have more selenium, the pathway of arachidonic acid metabolism leading to what are called these eicosanoids, which are all these prostaglandin E2, thromboxanes, all this, they are called eicosanoids. These bioactive lipids now switch towards this pathway versus this pathway. It's like your politicians. You give them more money, they'll switch this side. You give them less money, they'll switch that side. Just like that. Okay? Politicians are politicians. They're the same everywhere. Anyway, so these mediators then activate different transcription factors and then turn on and off the genes. And we use a technique called CHIP. You may have heard this, CHIP. It's called chromatin immunoprecipitation. Have you heard this? What we are trying to do, transcription factors. These transcription factors are binding to the genes at the promoters, correct? Yes? Now, when they are binding to the promoters, you can actually freeze that interaction and pull the promoter with the transcription factor bound there using an antibody, and then you can do a PCR to see if the transcription factor is binding to the DNA or not. And you can do it with a qPCR or qRT-PCR, actually not an RT-PCR, but qPCR, and quantitatively measure how much of transcription factor is binding at a site. And now you can do it at a whole genome level, and we do what is called ChIP-seq. Okay, you have heard of next generation sequencing, NGS. Just like that, you can do ChIP-seq. Okay, it's now become obsolete pretty much. In the ChIP-seq, we can actually now profile all genes that are bound or not bound in any disease condition. And what we see here is a simple PCR of one of the chips to show you that in this gut-derived cells, macrophages, there was an increase in some of the PPR gamma binding genes to a certain promoter, which is regulated by this pathway. You don't really need to know that, but just to kind of say that PPR gamma is a transcription factor that is activated by this side of the pathway, and that is now activated by this. Now, these prostaglandins are very important because they can bind to different proteins because of their structure. And you can see these prostaglandin J2 have a very different structure compared to all prostaglandins. And this is where chemistry comes very important. And this particular carbon is highly electrophilic. So what it is it is looking for electrons to go and pair, looking for nucleophiles. This particular carbon, you have heard of electrophiles and nucleophiles, correct? So this particular carbon here, okay, it's called C9 carbon, is extremely electrophilic. What it does is it will go and bind to certain cysteines as shown here. This is a cysteine in the active site of a protein that we reported many years ago. where it goes and binds covalently, and you can actually pull that down as a complex. Now it does that to many other proteins, and it can modulate signaling kinases, chromatin dynamics, oxidative stress, transcription factors, many things. So it has multiple targets in cells. And as a result of all this, what happens is, because of the production of these prostaglandins, it will now take a cell that is highly inflamed and turn it to an anti-inflammatory cell. Okay? It is sort of taking a rowdy cell and making it a nice saint cell. Let's put it that way. Okay? So that's what it does. And if you look at macrophages, just like T cells, macrophages also come in different flavors. There are macrophages that are highly inflammatory. It is called M1 macrophages. And that's what you see in disease in colitis. They are very highly inflammatory. But then, if you give them more selenium, they increase this endogenous bioactive lipid, PGJ2, and they become M2-like. These M2 cells are highly resolving, and they are not inflammatory. They've actually switched off many genes, and they are now resolving cells. So these cells start to come up. And it actually helps because these cells help resolve the inflammation. Okay? And that actually helps in resolving colitis. So what happens is, to make a long story short, when you give selenium, it switches the pathways in macrophages, particularly in the gut, in your intestine, and switches them to make more and more of this resolving mediators, which activate PPR gamma and then lead to anti-inflammation and wound healing and helps in that process. Okay. So I am. You want me to show some, or shall I stop? A few more slides. Are you okay with that? Yes. All right. So okay. So we decided to. I'm skipping the parasite infection and instead keep with the colitis flow of things. So we decided to do. This was we didn't do this actually. I'm sorry. This was done by our colleague at University of Pennsylvania, Gary Wu, who actually, instead of giving this selenium, he decided to give a drug that is used for diabetes. And that drug is called rosiglitazone. Okay. It's a drug given for diabetes to reduce, of course, the hyperglycemia, but then increase fatty acid oxidation. And what he did was, he gave mice the drug instead of selenium, and he actually had the same outcome. Now, the problem with that drug is, it is now out of the US market. Even in India, they have stopped giving the rosiglitazone. Pioglitazone is another drug of the same class, a next generation, but again, that is not given much. We are back to our old drug called metformin, okay, which has multiple targets. Anyway, so this PPR gamma, you can't use the drug because of liver toxicity. So we are back to selenium again. Okay, but where you can actually make your own cells generate the ligands for PPR gamma and help resolve inflammation. So we decided to take, one of the things that we first tested was, how important are macrophages in the inflammation? So the first question we asked was, are macrophages important? So if you use a drug called clotronate, okay, this is a drug that you inject into the mice, it will kill all the macrophages. Okay? And what happens is, if you kill all the macrophages, even if you give them selenium, it will not resolve the disease. So what this told us was that macrophages are important. And selenium in macrophages is important. Okay? Then we said, what if we knock out the tRNA that I showed you and use that model where macrophages don't make selenium proteins at all, but you feed them high selenium. Now they eat a lot of selenium, but they don't make selenium proteins. Now, do those mice actually resolve colitis or not? They don't resolve either. even if you give them high selenium, they don't resolve. Which tells you, not only macrophages are important, selenium is important, but then, in addition to selenium, selenium proteins are important. Okay? So then we decided to understand the mechanisms. How is it controlling? So macrophages are so important. How is selenium proteins controlling macrophages? So we did a metabolomics study. This is published in JBC last year, where we profiled all kinds of metabolites. Okay, it was called a shotgun method of metabolomics, and when we did that, we actually were able to use. This is where bioinformatics, advanced programming, and R language or R scripts are important. How many of you know R scripts? One. It's a tool, I think, which will be very helpful if you can learn that. Okay? See if you can learn. It's open source. You can actually do it. So see if you can do R, if you can learn R. You should be able to. You're all very smart. So you should do it. Okay? If you can do that, you can actually analyze very complex data sets using R. So see if you can do it. So using R, we were able to clearly parse out the differences and show how selenium and selenium proteins are impacting metabolism. So what we found was, making a big story short and simple, we found that when there is selenium deficiency, glycolysis increases and TCA cycle shuts down. So, as you know, glycolysis is very inefficient in producing energy. So what happens, the cell is cranking up a lot of energy or metabolites and of course glucose and others, but it's not making energy so much. It's wasting a lot. Whereas when you add selenium, you now decrease glycolysis and increase TCA cycle to its benefit. Okay, this is exactly what Warburg phenomenon is. You heard of Warburg effect in cancer? What Dr. Warburg in 1930s discovered, we have now epithelium in the colonic epithelium. You have epithelial cells, right? And those epithelial cells are nicely joined. Okay, like you have put some araldite or something and they are all nicely joined, and that is because of these proteins called occludin, and there are proteins called ZO1 or zona occludens protein. Now these proteins are very important in your gut, because if you delete any of these proteins or if something happens, you will now make your gut very permeable to bacteria. You know that you have more bacteria in your gut than your number of cells in your entire body. Right? You have more number of bacteria. Right? You have heard of the term microbiome. Microbiome is important. Even, to signal to the brain, microbiome is important. Right? So, any changes in the gut, and if you have this particular change where you have now disrupted the epithelial barrier, you have made more space, so the bacteria will go through and you may have an infection. So we monitored that particular protein, ZO1, in the epithelium, and we found that selenium actually helps regulate that protein. Not only that, we also gave mice a drug that's nothing but a fluorescinated molecule through the diet, and we were trying to see how much of it comes in the circulation, in the blood, peripheral blood. So that told us that if there is barrier integrity is really very strong, that drug will not come into your blood circulation, which means it's not absorbed. But if the barrier integrity is loose, it will go through and it will come in the blood. And we were able to monitor it because it has a tag called FITC or fluorescein. So fluorescein is extremely fluorescent and we can measure it. So by doing that, we were able to see that selenium actually regulates that too. And deficiency actually increases, even though this is with a slightly different inhibitor, but this is what happened. So selenium deficiency actually caused a lot of disintegrating of that variable. Okay? So what I told you was that selenium is a micronutrient, acts through these proteins called selenoproteins. And the molecular biology is very interesting. It uses a UGA codon to code and then incorporates selenocysteine. That is important. Selenoproteins are important, particularly in immune cells, to keep inflammation low and help in metabolic reprogramming through multiple different pathways and helping general health. Okay. With that, I will thank all my students, my research staff, many different people at my institution, as well as funding sources. And I will leave you with this. I think I have shown this in the past, but you may have seen this. Time magazine recognized inflammation as the secret killer for many diseases: heart attacks, cancer, Alzheimer's, GI diseases, and so on. Right? And the secret is here. Is selenium. Okay. Any questions? Thank you so much once again for the invitation. Wonderful to talk to you. Thanks. Happy to answer any questions. I always tell my students there is nothing called a dumb question. Every question is important. Yes.
Is there any downsides of using selenium to treat inflammation? If no, then why don't we replace our steroidal drugs with selenium? Because we know that it has a lot of downfalls like osteoporosis when we use steroids.
Very good question. The question is, are there any downsides to using selenium? Yes, there are. The issue is toxicity. So, one of the things, and we are all working towards trying to do exactly that, to see if we can replace steroids with selenium. So to get there, we want to make sure we do enough testing to recommend that. At this time, we know that selenium acts in a certain, there is a therapeutic dose that you can give. But if you exceed, it can become toxic. But again, the toxicity goes up, it's pretty high. You have to go really very high to get there. But still, you have to be careful. And then there are other issues with mutations. Remember, I showed you this particular slide of this one, right? This particular slide. So there are some mutations in people where, despite giving selenium, these proteins, I don't know about India, but I think elsewhere, you can go to a general store over the counter and you can get it. So people are doing that. And some people actually see benefits, and some don't. And that is probably because of all the mutations. Good question. Any other question?
So, basically, emotional imbalance, the person who gets frustration from continuously because of the nutrients imbalance also a factor, one of the factors.
Sorry.
Emotions, the feeling very sad or feeling happy or feeling angry, most of them is because of nutrients imbalance also can cause kind of multiple emotions that because of nutrients.
Yes. So, the question is, nutritional deficiencies or diseases in general do have a psychological bearing, right? Make you feel, emotionally down. It can happen. And there are some reports where selenium is thought to elevate the mood and help make people happy. But I don't know how that happens, but it is known that way. It may treat that as well. You're asking me if it can work as an antidepressant. There are some reports, but I'm unless we have strong evidence, scientific evidence, I'm not very comfortable in saying that it can do it.
Through genetics, we used to say also, like our grand, some like if you some members have short temper, some members are very emotional or everything. It's because through genetics they have even their father used to react for same thing like that. Even the grandfather used to react. It is because of genetics the environment they have been grown, the way of reacting the things like that. Maybe the short tempered or highly emotional or highly sensitive of reacting of some things happened to them. It's because of genetics or because of environment, what in which environment have been grown.
Maybe both. Genetics and nature and nurture, both. Yeah.
But we can say that genetics is a matter because he acted the same thing because maybe the father has same kind of reactions. The son can be reacted like that only.
Possible. Yeah. Maybe. I don't know the answer to the question. Any other questions? Yes.
So is selenium deficiency in infants?
Yes. 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 Selenium deficiency in infants is known. Yes. Particularly in breastfeeding mothers, where selenium intake is low. And in fact, what we do is, I wrote a review several years ago on recommendations for infants to elderly people, including pregnant women. And we saw that based on review of literature, it's found that pregnant women and nursing women actually need to increase selenium. That helps in not only increasing selenium in the milk, but also helping, because you know that many of the antibodies can be transferred. And for antibodies to fold correctly, antibodies need to fold properly. For that folding to occur, you need some selenium proteins. There is one selenium protein called selenium F, which is thought to be a gatekeeper for antibody folding. And that actually might help in the antibody folding process. So you have to increase selenium in nursing women and pregnant women.
How do we maintain the right selenium diet naturally? What kind of nutrition do we take?
Yeah. How do you maintain right selenium concentration? Yes. So, yeah, it's not, you cannot test it like a prick test for your blood glucose. Unfortunately, we don't have that kind of technology yet. We have to go through what is called ICDMS or mass spectrometry based. So, best is to actually eat a healthy diet and increase. You don't have to eat non-vegetarian food to sleep, because you can still get selenium from eating cruciferous vegetables, and you can also get selenium from garlic and broccoli. And I know nowadays broccoli is pretty popular. Broccoli has a good amount of selenium as well, so you can take those, and it should give you enough selenium.
We saw that selenium is given as supplements for hypothyroidism. So is it equal for even hypothyroidism?
No, usually not.
Are there any studies being made to reverse hypothyroidism? Because we have seen that if a patient gets can it be reversed?
Unfortunately, that's what it is, because of many reasons. So you take some of this till you kind of bring up your T3 levels, and yes, you're done. Right? Yes, if you know exactly where the problem is, then that's where personalized medicine comes into effect, and you can actually treat it. You can improve it. But you may have heard already that with the CRISPR technology, when it came out, CRISPR works incredibly very nicely in any system you can think of. You can take a brain cell and add or delete a protein and do things like we can do a lot of things. It's like almost like voodoo magic, you know. But you have to be careful. Genetic and manipulation of human embryos like that happened in China. You have to be very careful because we don't know what could be the long-term consequences. Because we know if you engineer a certain mutation or correct a mutation by CRISPR, you don't know if that could reverse over a long period of time if you don't keep sufficient pressure or whatever. If that happens, then suddenly a person may have more problems. So genetic manipulation is still there are a lot of ethical issues. So unfortunately, it will take a long time.
So also cancer patients have the ability to have cervical cancer. So due to radiation, gut is damaged. So can selenium supplements help reduce that this thing?
It is a possibility.
Yes. Yeah, I think before, I think selenium might be able to help. Yes. But I think it is better not to do it blindly. It is better to consult an endocrinologist or a clinical nutritionist to make sure there is selenium deficiency. If there is selenium deficiency, perhaps it will definitely benefit. Yes.
Sir, in case of extreme selenium deficiency, what is the most efficient way to give selenium into the person's body?
Yeah, so in terms of extreme selenium deficiency, you can give them inorganic selenite. Okay.
So, an oral supplement.
Yes, oral supplement, 200 micrograms mcg per day should help.
Is this the same for kids and adults, or is there?
Kids, we decrease it. Yeah. Kids, we decrease the dosage. I think it's about 60 micrograms or so, up to about 2 years. And 2 to 10 years, I think there is a different range. And then you increase.
Thank you.
So is selenium recommended for other severe patients like
Yeah, cardiovascular disease and selenium, right? Yes, there are some reports, particularly to help with lipid levels, because finally it is the bone cells of oxidized fatty acids and lipids. So those, and that happens because macrophages are like phagocytes, and they take up particularly these oxidized lipids. To keep those oxidized lipids low, selenium will help. Definitely help in keeping them low. So, like maybe.
because they could take other tablets also.
Yes.
There might be side effects. Is that?
Yes, that's actually a very good point. You have to be careful when you take multiple different drugs and then combine it with selenium or anything else, or even take Ayurvedic medicine, because you will change the pharmacology. You will change how fast the drug is absorbed. But I'll give you one example. You all like Isabella, right? I don't think there is one person who says no. So, or you like sambar, right? Now, in sambar, you have put turmeric, right? Curcumin. And curcumin is such a, it's a wonderful molecule, has a lot of health benefits, no doubt. Now, amount of curcumin you take, you can dump yourself with a lot of curcumin. You only absorb a small fraction of it. Not just curcumin, but also a group of compounds called curcuminoids. You only absorb a little bit. Now, you add a little bit of black pepper to it. You see where I'm going? I'm making sambar. So if you add a little bit of black pepper, you increase increase your curcumin absorption significantly. Because black pepper has bicolor, is a compound. That increases some of the transporters which absorb. Now this is not just restricted to curcumin. If you take any other drug along with pepper, you will increase their levels too. So you may be taking a drug and now you take some sambar or eat sambar and you have pepper in there. You may actually increase the absorption of the drug. And that could be good or bad depending on the drug. Right? So this is where diet plays a very important role in how it can interact with your pharmacophores or pharmaceutical agents. So now you see interaction between drugs. And majority of your drugs are being developed from plants. Over 60 to 70% of the drugs currently that we use are all coming from plants. Good question. Any other questions? Or questions?
Sir,
Yes.
is irritable bowel syndrome same as this one or is it different?
Yeah, it is the same.
Same.
Yes, IBS.
That
Yeah,
is
it
also
is.
due to selenium deficiency.
Well, no, they are not all due to selenium deficiency. We don't know. We don't know what are the etiological
They
factors.
say there's no cure for it. Like every
That-
patient tries to show different.
It's a spectrum.
Yes.
Yes, it's a spectrum and we don't have a cure. And IBS is a general term for many diseases of the gut. IBD is one. Within IBD you have others like you have colitis, you have Crohn's disease. Yeah, but it's in the same family. Any other questions?
Last question.
Last question.
Recently I came to know that they have in US they have done survey that what is the life duration of vegetarians and non-vegetarians. They came to know that final conclusion that vegetarians have lifespan duration is more than non-vegetarians. On what basis the vegetarian has more lifespan?
All herbs are good.
All herbs are good. So, I think, I have seen some studies like that non-vegetarians versus vegetarians. I don't know. There is genetics. There is also how much you eat. Tasty food you may eat more unless you don't have self-control. Or it's just that you may eat stuff. Like for example, if you were to eat say products that have components of blood or heme. Now you're also overloading yourself with iron as well. Like for example, I have heard that people eat, they like liver, right? It's a delicacy apparently. So if that is really true, if you eat more liver, you are getting a lot of, you are getting hemoglobin, you are getting bile acids, you are getting a lot of other stuff. So that definitely has an impact on your microbiome. And your microbiome changes can impact many things. So it finally all boils down to microbiome, the bacteria. Because there are studies which have shown that, not to, again digressing from your point, they have shown that people who have certain diseases, particularly autism, autism is now very important, and it's a disease where we really don't have any cures. But what people have shown, particularly in Canada, a clinical study that was done, there is a link with microbiome, and what they have done is they have done fecal transplants. So what they have done is they have taken the fecal matter from healthy relatives staying in the same household as the autistic individual, and of course, process the fecal matter in such a way that you will not even realize that you are taking a fecal pellet through capsules. And then they have given that, and that has actually corrected. And what that tells you is you are now repopulating the microbiome by giving that. And so that repopulating the microbiome has changed the autism phenotype, at least given a lot of relief. Because if you see those kids with autism, it's really sad. And if you can correct them, it's a very simple thing. So they have done that. That study, I heard the person who was actually doing this study tell us about it. So it was very positive.
Even genetic defects, last question, genetic defect which cause the offsprings, like many syndromes, Down syndrome, many syndromes. Even the nutrients also plays a major role, sir.
Yes. Questions? If not, thank you all. Thanks a lot.
And I'd like to thank Dr. Sandeep Prabhu sir for delivering such a thought-provoking and informative session. Now I'd like to call upon Dr. Haraprasad to give away the memento as a token of gratitude. Also, I'd like to thank everyone present over here and patiently listening. Thank you, Madam.
Beautiful.